Welcome back to the Destroy Duchenne podcast, a podcast aimed at telling the stories of those with a relationship to Duchenne. My name is Stacy. I'm a patient with Duchenne, the founder of Destroy Duchenne, an author, and I'm a consultant on behalf of Satellos Bioscience, as well as other biotech companies. Today I have with us co-founder and chief discovery officer of Satellos Bioscience, Dr. Michael Rudnicki. Dr. Rudnicki is also the scientific director of the Canadian Stem Cell Network, director of the regenerative medicine program at the Ottawa Hospital Research Institute, professor in the Faculty of Medicine at the University of Ottawa. Welcome to the Destroy Duchenne podcast. So, Dr. Rudnicki, it's a pleasure to have you here. And I first want to start off about your background. So, what is your background and what drew you to this field of science? Have you always been curious about this or biological sciences since you were a child? Or tell us about that.
Yeah, well, first of all, thanks very much having me on your podcast, Elijah. Uh, and it's great to see you again. So, yeah, growing up, my brother and I were, as smaller kids, became really, I think, first interested in science by, watching some television shows about science. I wanted to be a marine biologist initially from watching Cousteau's show uh, about divers and underwater fauna and that kind of thing. And then in high school, I read The Selfish Gene by Dawkins, and then The Double Helix, and that got me really interested in genetics, DNA, and then molecular biology. And so when I went off to university, I went into a molecular biology program. So I was trained as an undergraduate as a molecular biologist, um, genes, gene expression, DNA, et cetera, et cetera. And I had some really inspiring professors. Um, so I decided to go to grad school and try to understand how things work. And I went to work in a lab whose head, Michael McBurney, he had trained with James Till and Ernst McCullough, the two Canadian scientists who were the very first to demonstrate that stem cells existed. And I was working on embryonal carcinoma cells, which is what people worked on before there were embryonic stem cells, but they're very similar, but they come from a tumor. Working on muscle differentiation and control of gene expression through muscle differentiation. So that's how I got into science.
So, you definitely had some pretty significant teachers in your life that you studied under. Then that's amazing, and so that's pretty cool to hear. I hear a lot of stories about people getting drawn to science from reading certain books. Some of the same books that you talked about now. That's pretty cool to hear that you have a similar story. When it comes to Duchenne, right? When did you start researching it?
Well, so I was supported by two different types of studentships as a student. Either initially by cancer type studentships, and then later by muscular dystrophy type studentships from from a foundation supporting muscular dystrophy research in Canada, uh, and that made me aware of patients, the disease. I met some patients, and through fundraisers and that kind of thing, and met their families, and that really puts a real dimension on this disease. And so it becomes to mean more and more, having an impact, trying to have your research translated so that you can maybe change the world a little bit.
Do you think it's important to do science that will have a direct impact on people's lives? Not just doing it to learn things, but doing it that translates into something that is
Yeah,
meaningful to you.
I think what happens, science for its own sake has to be done because it's a kind of a false dichotomy applied versus discovery research. But as the arc of one's career, one becomes drawn to more translational applied problems. You want to see your discoveries get out there into the world, and that's a good, I think, healthy thing. And so people who start off as curiosity-driven basic scientists often move over and move along to moving their stuff out into the real world through the arc of their career. And that's exactly what sort of the pattern that happened to be. As I had my own kids and raised my family, the understanding of what Duchenne patients go through, what happens to their families, becomes really motivating to if you think you can make a difference, to try to get it that.
What is it about patients with Duchenne that you noticed that made you, I don't know, I guess be touched by it, that you wanted to really help them? Like, what did you notice about their lives that you're like, okay, I want to go into this and make a difference? Because you did mention that you met some patients with Duchenne and that shaped your interest in studying it.
These are little kids, right, and young adults, and it's a as you know, it's an awful disease, and with tragic consequences, and it's affects the whole family.
Absolutely, absolutely. That's something I always talk about, is that Duchenne doesn't just impact the person, but everybody around them. Right? It's like a bomb that has a radius, radius blast that just
Yep.
affects everything it sees. But going into Duchenne, can you explain what causes it?
Yeah. So it's an inherited disease. It's the disease gene that's deleted or mutated is called dystrophin. It's the largest gene in our body, in our genome. It's located on the X chromosome, and females have two copies. And so if they have a mutation in one gene, they're unaffected. Males have one copy because it's on the 6 chromosome. The other chromosome is a Y. So if they have a mutation, they have the full-blown disease. So that's why it's almost exclusively in males. The odds of a female having two copies of a mutated gene are very, very small. But it does happen rarely. Um, and it's the most frequent kind of muscular dystrophy, somewhere between 1 in 3,500 and 1 in 4,000 births. It's a big gene, so almost half the mutations are new mutations. So it's a big target for mutagenesis. And the protein dystrophin has been known for a long time to be expressed in differentiated muscle fibers, as well as many other cell types of the body, but where its effects are really felt are in skeletal muscle, where it was long been believed and understood to be a kind of a shock absorber in muscle, connecting the proteins inside of the muscle fiber that control contraction and strength, and the actin tubulin networks to the outside of the cell to keep everything in register and prevent damage. And the absence of dystrophin, there's a loss of structural integrity, and when muscle contracts, there's tears in the membrane that causes influx of calcium, outflux of stuff that's required for muscle fiber function, muscle damage, and rounds of degeneration and regeneration that results in loss of muscle fibers and replacement of the tissue with scar tissue and fatty tissue and loss of strength. Now we understand there's other issues in muscle, not just the structural integrity. And that's the stem cell aspect.
Yeah. Let's talk about that. So you made a huge discovery, which was published in the journal of Nature Medicine, that dystrophin is actually present in muscle satellite cells,
Okay.
which are the muscle stem cells. Walk us through that. Talk to us about that. Very fascinating with that.
Yeah, so that was one of those things that happened in the lab that we had an observation. We've been studying asymmetric stem cell division, and I can explain that perhaps later, or maybe I should explain it now. So when a stem cell divides, and this is many different types of stem cells, including muscle stem cells, it can divide in two ways. It can divide to make two stem cells. That's called a symmetric division because it's making two identical daughter cells. Or it can divide asymmetrically in a 90-degree angle to the muscle fiber to give rise to a cell. One cell remains a stem cell, and the other cell becomes a cell that's going to go on, proliferate, and then differentiate and in effect repair the tissue. and that's called an asymmetric division. Sorry for the science lesson. The and what we were doing control experiments, and we were just looking at in muscle fibers isolated from mice with Duchenne muscular dystrophy, and we noted that the baseline level was really wonky. There was a dramatic loss of the expected numbers of asymmetric divisions. So we scratched that heads. Why is that? And so is it environmental, maybe caused by inflammation, signaling, giving abnormal signalings to these? But these are now ex vivo, and we're seeing this. Or is it something about the satellite cell itself? And I wondered, well, maybe dystrophin is expressed in satellite cells. The field had a dogma that it was not in there at all. And that's because if you put satellite cells into petri dishes and grow them, you get myoblasts, and they don't express dystrophin at all. So it was not thought to be a feature of these cells. But what we found at the RNA level, there was super high levels of full-length dystrophin. And when we looked at the cells as they were getting ready to divide, the protein was turned on and was expressed at a very high level. But it wasn't expressed around the cell. It was expressed at a cap and polarized in that cell. And so the penny dropped. We're looking at a new role for dystrophin as a signaling molecule, regulating polarity, positional identity, and impacting asymmetric division. So it led to a whole series of experiments that I think really provided a paradigm shift in our understanding of what drives the progressive nature of the disease. You have this ongoing muscle damage in fibers, and secondly, you have this impaired ability to repair that damage. And those two things together drives the disease forward in a progressive way.
Right, now that's incredibly significant. I mean, would you say then it's fair to call Duchenne a stem cell disease in the sense that that's where a huge part of the problem lies? Is that you need to fix
I,
that
I like
issue?
that, but a lot of people were, at least in the early days, were quite annoyed by that. but a colleague of mine, Peter Zammit in London, has written a couple of reviews or so where he's coined this term satellite cellopathy, and has classified different muscular dystrophies and whether or not satellite cells are affected. Duchenne is the penultimate example of that.
What does it feel like to be going up against all the dogma and making a discovery that challenges traditional knowledge for the longest of time to discover something that changes the way that we understand the disease? How does that feel? What is that like?
That's great fun. I mean, a lot of people are good scientists, like to engage in different ideas and discuss it out. So the dogmatists are a minority with an axe to grind, perhaps, and hopefully not too powerful. We were able to get our paper published. In worst case scenarios, I suppose that can lead to difficulty in getting stuff published, but if you go against the tide.
Okay. Yeah, but that's just an amazing discovery, and I'm very excited about it as a patient with Duchenne. So thank you for your work doing that. But moving on to my next question, there's been studies that show patients with DMD actually have an elevated number of satellite cells or muscle stem cells. Contrary to popular belief, which we just talked about, that regeneration is impaired to satellite cells becoming exhausted and then running out. Talk to us about that, and could this potentially mean that patients with DMD actually benefit more so from correcting satellite polarity, as the body
Yeah.
essentially has more material to build from?
So that term, a stem cell exhaustion, was coined by workers like Helen Blau, and that was because there seemed to be a lot of satellite cells, but they weren't repairing the tissue very well. So she hypothesized that through overuse that they were becoming pooped out, which was a reasonable hypothesis, but it was just an interpretation of what they knew at that time. That somehow morphed into depletion. And in many reviews around that time, and when scientists were giving talks or clinicians were giving talks, they were talking about depletion of satellite cells or muscle stem cells in DMD. But the data didn't say that at all. In fact, multiple published papers showed that there's elevated numbers of muscle stem cells, what's called a hyperplasia of stem cells in DMD. They're elevated 3 to 5-fold at all stages of the disease. And this is also true in animal models like mice and dogs. But it wasn't understood why, because if you put those cells into cell culture, they seem to differentiate pretty well, almost indistinguishable from normal cells. And what was the problem was this deficient asymmetric division. So rather than making one stem cell and one committed cell that would then go on and differentiate, they made 2 stem cells. So the numbers of stem cells was ratcheting up over time, rather than staying at normal levels. And so that's another example where dogma is trumping data in people's minds, because it made a nice story that stem cells are exhausted and a nice explanation, but it was incorrect.
So in terms of correcting it, since there is an elevated number of satellite cells, could that potentially mean that if you were able to correct that polarity issue that we see in DMD, which let me first give an analogy for the audience. You tell me if this is correct. The way I like to think about it is it's kind of like the Earth. You have a North Pole and you have a South Pole, and that creates that polarization. But within inside the satellite cell or the muscle stem cell, you have different proteins. One of those being dystrophin, which then creates that polarity with the other proteins, such as like part 3, which would be, let's say, the North Pole. And since you don't have dystrophin, you no longer have that South Pole. Now you're lacking that polarity, and so the cells aren't able to go through that asymmetric division. But
Yep.
if you're able to correct that polarity, then they would be able to do so. Would that be a fair analogy?
Absolutely. So these cells are wedged into a niche that's located between the muscle fiber and an overlying membrane. It's called a basal lamina. And the cells have different proteins adhering to the membrane versus other proteins on the other side adhering to the muscle fiber itself. And so those cells are polarized. They have an up and a down, and or a north and a south pole. And in the absence of dystrophin, they have a hard time figuring that out. And so in order to undergo an asymmetric division, they need to have that directional sense working. Otherwise, the pole of cell division becomes randomized, and the proportion of asymmetric divisions drops quite dramatically. So that's a very nice analogy.
Awesome, awesome. So then I guess going back to the elevated number of satellite cells, since there is more satellite cells to work with, if you were able to correct that polarity, couldn't that potentially be a benefit? Couldn't the problem be somewhat of a solution towards Duchenne in the sense of fixing the impaired regeneration problem?
Absolutely. As long as you have muscle stem cells and you can mobilize to generate progenitors through asymmetric divisions, you can repair the tissue.
That's
So
awesome.
it's yes, what you say, I agree with. Yeah.
That gives a lot of hope. I think as patients to know that that is a possibility, and I think it's somewhat beautiful that the problem it created more satellite cells, but that actually could be a potential benefit for patients with the disease if you could fix this issue. Um, so, kind of moving on, which I think really gives a lot of credibility to your work and to what Satellos is doing, are these studies. So, tell us about the studies of patients who have seemed to have a disease-modifying gene and are able to still walk in their 20s and even 30s, and also tell us about the dogs and other animals that we see that have these
Yep.
escaper genes.
I'll start with the dog. This is work by Mariana Katz in Brazil and Lou Kunkel, who also discovered the dystrophin gene. He's runs a lab at Harvard Medical School. And she had found in her colony of Duchenne dogs a dog that had no dystrophin whatsoever, but is a normal dog. In fact, that dog is still alive. And so they did sequenced the genome of those dogs and looked for differences, that dog and other dogs that were like that, and found a mutation that caused a change in the level of a protein called jagged1. Jagged1 is a gene makes a cell surface protein that stimulates Notch signaling, and Notch is a receptor on satellite cells that regulates polarity. And they put that upregulated jagged1, or they arranged for zebrafish with Duchenne muscular dystrophy to have an upregulated jagged1 using transgenes, and they essentially fixed the disease by enhancing regeneration. And those dogs have no heart problems, and they're pretty healthy, even though implying that by repairing the skeletal muscle tissue makes the heart, puts less stress on the heart. I think that's a reasonable interpretation. And then a few years after that, Kevin Flanagan published a paper of patients who have absolutely no dystrophin who have prolonged ambulation. And they found mutations in a number of different genes. And one of the mutations they hypothesized leads to an upregulation of a protein called PAR-D6G, which is part of the polarity complex, together with PAR3. And those patients walk past 30. So maybe 15 years more ambulation. And then Mayanna Katz and Lou Kunkel recently, Lou spoke about this on a YouTube broadcast. They identified mutations in Notch3, which is a Notch member expressed in satellite cells, muscle stem cells, that similarly leads to ambulation past them. Those patients are still alive, I believe. And there's two families that they found these mutations in, one in Spain and another one in Brazil. And in one of those families, there's two brothers with Duchenne. One of them has a Notch3 mutation, the other doesn't. And it was the one with the Notch3 mutation that continued to walk, whereas his brother was in a wheelchair from a very young age. So those experiments together implicate regeneration, polarity, and in particular the Notch signaling pathway in being important in amplifying the polarity signals in a muscle stem cell without dystrophin to rescue that loss of polarity and drive the repair processes and normalize the repair processes so they can keep up to some extent with the ongoing damage.
So to offer a simplification of that, essentially what you're saying is that because these patients were able to have a normal regenerative process of muscle tissue, they were able to still function a lot longer, even though they had no dystrophin, which is highly talked about within the Duchenne world. Is that correct?
Yeah, I mean, the muscle is still, histologically, if we look down the microscope at it, you can tell it's dystrophic muscle. But the damage and the loss is blunted, so that maybe you could think of it as more Becker's like, but it's returned towards normal, but it's not completely normal. But certainly the finding in dogs suggests that with the right intervention, we could do even better.
So we talked a lot about your research and things like that, but then that leads to Satellos. So what made you want to start Satellos, and how do you know the CEO and other co-founder, Frank Gleason?
Frank and I are old and close friends. We've worked together now for over 20 years. We first met when I stepped up to the assumed the role of CEO and scientific director of the Canadian Stem Cell Network. He was a member of the board. And then shortly after that, he became chair of the board. And he was a mentor. He helped, I was quite young and wet behind the ears and green in that role. And he helped me get up to speed. And him and I talked a lot. And one of the things that we together wanted to um, do as a new program on the network, and we both felt strongly about this, was to support through our research grants programs that would identify drugs that stimulate regeneration by acting on the stem cells that are in our body already, mobilizing them. And out of that program came some really successful drugs that allow amplification of hematopoietic stem cells, blood stem cells in bioreactors, for example. And there's other examples. But that um, that notion, that idea that drugs could be used to stimulate intrinsic regeneration has always been a vision on the part of both of us. And when my work began to uncover mechanisms that can modulate stem cells and in particular restore asymmetric division via pathways that we knew were druggable, um, and that it appeared to ameliorate disease progression in mouse models of DMD. That got us both very excited, and we wanted to push forward and spin out a new company to do that. And when we pitched this to the people at Bloom Burton, John Holyoke and Brian Bloom in particular, they both got very, very excited by this idea. Whereas everyone else that we talked to just showed us the door and said, here's your hat. What's your hurry? Because gene therapy was going to cure DMD. Everyone felt that. Everyone was convinced of that. It's done. It's finished. And so we were able to form a little company and raising a bit of money at a time, bootstrapping. We were running on fumes a few times. We've been able to move forward, and I think we now have a real company. So it's very, very exciting times at Satellos.
And what is the vision of it? You kind of touched on like being able to stimulate, um, you know, stem cells from within intrinsically. But I mean, the slogan is to rebuild muscle from within, right? And I think that's a beautiful slogan that kind of summarizes the mission
Yeah,
perfectly. But what would you say the vision is of Satellos?
just that, rebuild from within, and that vision, I think, is will really, if when applied broadly in multiple contexts against multiple targets, I think will really bring a maturity to the field of regenerative medicine. So regenerative medicine now is widely felt to be transplantations of cells of tissues, bioengineered valves, that kind of thing. But it's also stimulating intrinsic repair by targeting the mechanisms that we and others these mechanisms that control stem cell function in all kinds of different places. And I still feel that that's really we will have arrived at the new age of regenerative medicine when we have a pharmacopoeia, different drugs that target different stem cells in different tissues in different contexts and for different diseases. I really think that's a very exciting way to go forward. That doesn't mean that we shouldn't transplant cells. That's still effective, important set of therapies for moving forward. But we also need to be able to stimulate interest.
Right, because there's a huge benefit to that. A lot of people they look at stem cell therapies where you have to transplant it, and that has a lot of issues, especially in the context of muscle,
Yes.
because it's hard to get it to all the different locations. But if you could just stimulate it from within the body, then your body will naturally get it to everywhere where it's supposed to go, and that would be the benefit of it, right? That's the benefit of doing it from within.
Yeah. So, for bone marrow transplant, you can regenerate an entire blood-making system from transplanting a single blood-forming stem cell. It goes to the bone marrow and sets up shop and repopulates that niche, and away you go. For muscle, you need billions and billions of stem cells, and because they're highly localized. But if you put them into the blood system, they can't get to muscle. They're blocked. So you have to inject them. So how do you inject billions and billions of cells into over 600 different muscles? Some of them really hard to get to inside of our bodies and around our spinal cord. And, it's a real challenge, of course, to do direct intramuscular injections of stem cells. And then how do you get those stem cells? We can't grow them in bioreactors. We don't have the technology. We can grow myoblasts, but they don't transplant. So it's right now with the current technology, there's no solution. And so,
Yeah, not to mention, it would be pretty painful for the patient to have an intramuscular injection all over their body. That's a shot basically everywhere. So
it's a
I
lot
like
of
your
needles.
approach
That's a
better.
lot of needles. It's a lot of needles.
Yeah, yeah. So tell us about this novel therapeutic approach Satellos is developing to treat DMD. And I have a few questions to follow up about it, but tell us about what we're doing for DMD.
So we did a focused genetic screen by um, uh, looking at about 300 potential targets in muscle stem cells to explore whether they had a role in regulating polarity. And we were both looking at pathway identification and druggable targets in uh, those pathways. And one of the hits that flew through the secondary screens with flying colors was this protein called AC1, AAK1, which is the um, mammalian version of a fruit fly gene called NAC, or NUM associated kinase. And what NAC does in flies, it has a very well established and documented role in regulating asymmetric division by modulating notch signaling. And because we know notch signaling is important from the work from Mariana Katz and Luke Kunkel and others, that got us quite excited. And this is a kinase, so it's very druggable. It's a challenge drugging anything. But James Jasquith, our medicinal chemist, who is brilliant, by the way, he is absolutely brilliant guy, in a very logical series of molecules, came up with a very potent drug that has high affinity. It's very clean. It doesn't hit anything else, so it has high specificity, and we believe is going to be safe. So we're making a pill that would mobilize your stem cells, stimulate asymmetric division. This drug doesn't stimulate stem cell proliferation; it stimulates asymmetric division. So if anything, it's stimulating differentiation and the formation of cells that can differentiate. In our animal models, we see evidence for enhanced regeneration and improved outcomes, improved force generation, and so on. So it's really a innovative, radical, totally different way of approaching this, uh, and um, and this is an approach that conceivably we believe that any Duchenne patient could take. It doesn't depend on your mutation. It doesn't depend whether or not you have antibodies to AA. And as long as you have stem cells, it should have some positive effect. So we're very excited to move this towards the clinic.
No, I think it's amazing that it has a universal, a potential universal benefit to all patients. Your exon, your mutation status, your ambulatory status, potentially even doesn't even matter in terms of if this will work or not for you. And I just think that's really amazing. And also, too, you touched on that it's it's a pill. You could take this orally. That's a really big deal for patients, and it just almost seems like, wow, I could have you can have you can potentially create that effect by just a pill. It almost sounds um, almost too good to be true, but I mean, science is the science. Speaking of treatments, how would you define a successful treatment for DMD?
You know, speaking to patients over the years, uh, anything that improves the quality of life uh, would be a success. We're not talking about a cure at this point because we have no idea how until we get into humans what kind of consequences there'll be, and there's no cure in sight for any treatment at this point. Uh, um, but an improved quality of life, and some patients have told me, man, if I could use the mouse on my computer for a few more years, that would really be great, or be able to eat my meals, handle a knife and fork for a few more years, that would be awesome. Or be able to fly in a plane to Hawaii, that'd be awesome. So the expectations of the patients are not over the top. But what we hope to do is we hope, when we believe that we'll be able to extend ambulation and maybe better for several years or more.
Um, that's really exciting. So, question about your career. What was it like to be admitted as a fellow into the Royal Society and also having the chance to sign your name in the charter book next to people like Charles Darwin?
Oh wow, it was a tremendous honor. Just reflecting about it, it still gives me goosebumps. It was really quite a highlight of my career. My wife and I went over to London together. Almost didn't make it because of bad weather and flights cancellations, but we made it in the nick of time. And their headquarters is downtown London. It's a old marble building that was built before the Second World War, stuffed with artifacts, paintings, scientific instruments behind glass cases, busts of famous scientists. Their library has a collection of books, first edition, Origin of the Species kind of thing. It was hard not to put a few of those in my back pocket, try to sneak out the door, but I didn't. Um, and Oh, it was a tremendous honor. And then they have a ceremony where they welcome you. You get to sign this book, which is this ginormous volume that's I don't know, 2 feet by 3 feet. No, it's not quite that big, and it's with big brass binding and a big cover that they open up, and it's vellum pages, which is sheepskin, I think, that's been scraped and flattened. And on the first page is the uh, huge signature of Charles II. He writes founder for the Royal Society. It was formed in 1660. It was the first scientific society. They published the first journal. They held the first meetings of scientists to compare their discovery, and has Isaac Newton's signature, Charles Darwin's signature, and on and on and on. And so it was so exciting to get to write my name. I had to practice to remember how to write with a fountain pen so I wouldn't make a mess of it. Only a few years ago they did quills, and so that was very difficult to use. But I think probably about 10% of the people having to sign the book botch their signatures because they don't know how to write with a fountain pen, even though we practice. And then the other thing is, you want to write your signature so it's kind of legible. Most of us sort of just do a scrawl, So I had to practice so you could actually read it. Uh, and but And then they give you a scroll to take home and frame and that kind of thing. But I'm now involved with some of their different committees and that kind of stuff. So they are making me work. But man, it's a tremendous honor. It was a real gas.
No, I think it's super cool, and it's just really cool to be able to talk to you for somebody that is inside the Royal Society, and hearing about that experience is really really cool. Would you say that's one of your greatest career achievements, or is there
Oh
something
yeah,
else you would say is even better
absolutely.
than that?
Absolutely. Yeah.
Absolutely. Okay.
Yeah.
And then last question, what are you looking forward to in terms of the future for the Duchenne community?
In terms of the community, I certainly think what's really important is to have a relationship with the community as equals, as in a thoughtful, compassionate way, and a dialogue, an ongoing dialogue about simple things like design of the clinical trial, and getting input at all levels, and work with the community to ensure that what we're doing at Satellos is being done in a compassionate, effective, and we're treating human beings, so we're not, these are not lab animals, right? We have to care about uh, the people we're trying to help, and we do. I think that's really uh, part and parcel of who we are at the company, and it reflects the values of Frank and myself.
That's something that I really appreciate, and that's something I noticed really early on when I first came across Satellos in my relationship with Satellos, is how much you guys do care about the patient and trying to design trials and get patient input and, just that level of care for patients that really matters. And I really think that when you put the patient first, good things follow from that. So that is definitely recognized and appreciated as a patient. But Dr. Rudnicki, thank you so much for being on the Destroy Duchenne podcast and for telling us all about Satellos and the work you're doing and the research you're doing. I think it's just tremendous, and it gives me a lot of hope that things are going to get better with this disease. So thank you so much for being on.
And I want to thank you, Elijah, for the opportunity to be on your podcast, and also to congratulate you on all the great works you're doing.
Thank you so much.